To grow and multiply, cancer cells require substantial amounts of fuel. One of their primary energy sources is glutamine, an amino acid they consume to rapidly build DNA and proteins. Because these cells rely so heavily on this specific nutrient, oncologists have a clear target. By administering drugs that cut off the patient's glutamine supply, doctors attempt to starve the tumor to death. But clinical outcomes rarely align perfectly with this theory. When the glutamine supply dries up, many tumors manage to survive by rerouting their metabolism to consume alternative energy sources. At the same time doctors are fighting the tumor internally, patients are dealing with the physical toll of chemotherapy. One of the most distressing side effects is severe hair thinning, which drives many to look for their own cosmetic solutions. To treat this hair loss, patients frequently turn to over-the-counter supplements, specifically high doses of vitamin B7, more commonly known as biotin. Because patients view these vitamins as harmless wellness products rather than active medications, they rarely mention them during appointments. Surveys show that only 6% of patients report using these supplements to their oncologists. This clinical silence creates a two-front blind spot. It introduces a chemical variable that can skew a patient's data and provide the tumor with a metabolic bypass that doctors may never see. The first threat biotin poses occurs inside the testing lab. Oncologists rely on routine blood tests to monitor whether a cancer is in remission or if treatment plans need to be adjusted. These tests use specific chemical assays to detect cancer biomarkers. However, excess biotin circulating in a patient's blood chemically interferes with the binding mechanisms these laboratory assays use to read the sample. As a result, the tests produce inaccurate numbers. In prostate cancer, high biotin levels can cause falsely lowered prostate-specific antigen, or PSA. In breast cancer, it can heavily skew estradiol results. This interference creates a diagnostic illusion. The lab results may show a tumor is shrinking when it is actually growing, giving doctors a false sense of security while a tumor secretly returns. The second threat is much more direct, and it occurs at the cellular level. Researchers at the University of Lausanne recently mapped exactly how tumors survive when their primary food supply is cut off. This diagram of a tumor cell reveals that when glutamine levels drop, tumors adapt by pulling carbon from pyruvate, a small molecule created when the body breaks down sugar. To process this alternative fuel, the tumor cell relies on a specific mitochondrial enzyme called pyruvate carboxylase. This enzyme acts as the backup generator, taking in the pyruvate to keep the cell alive. But there is a mechanical catch. On its own, this enzyme is completely inactive. It sits at the entry point of the new fuel pipeline, unable to process the incoming energy. For the enzyme to activate, it requires a specific binding partner. Vitamin B7, biotin, is the mandatory cofactor that must attach to the enzyme to trigger its activity and restore the flow of energy to the cell. This is the intersection where a patient's hair loss regimen meets their tumor's survival strategy. By taking these supplements, they are unwittingly providing the exact chemical requirement the tumor needs to restart its energy flow. In their effort to recover from the cosmetic damage of chemotherapy, the patient inadvertently grants the tumor the metabolic means to survive the starvation strategy. However, this metabolic escape route has a natural vulnerability hidden in the patient's own genetics. Healthy cells rely on a specific gene called FBXW7 to manage the daily disposal of excess proteins. In certain cancers, this gene frequently mutates. When that happens, the cellular machinery breaks down, losing its ability to manufacture the pyruvate carboxylase enzyme. Without that enzyme present, the metabolic bypass fails. Even if the bloodstream is flooded with biotin, the vitamin has no partner to bind to, and the backup generator never turns on. A mutated FBXW7 gene physically traps the tumor. Unable to process alternative fuels, the cancer remains locked in its original glutamine addiction, stripping away its flexibility and making it highly vulnerable to targeted starvation. Because tumors are remarkably flexible, trying to eradicate them by cutting off a single food source leaves too much room for escape. Drug developers are now designing dual-target strategies. Future therapies must block the tumor's glutamine supply while simultaneously restricting its ability to capture biotin from the blood. At the same time, doctors can use genetic profiling to identify patients who already have the FBXWX7 mutation, allowing them to confidently deploy aggressive starvation therapies against tumors that lack a backup plan. But before these new drugs reach the clinic, the immediate reality of patient care must change. Patients must treat every over-the-counter supplement as an active chemical intervention and disclose everything they take to their oncology team. We often assume vitamins are universally healthy, but understanding their precise mechanical function reveals the truth. Introducing the wrong nutrient at the wrong time can be the difference between curing a patient and feeding their disease.